In this video, we're going to talk about the polymerase chain reaction in transgenic organisms and gene therapy. Alright, so let's say you have your DNA sequence that you want to clone, or you have your sequence already into a vector, or you are a detective and you find a spot of blood and you want to amplify the DNA to see whose blood that it belongs to. You can do all of that and much more using the polymerase chain reaction, or PCR. PCR is a technique to amplify DNA, and the way it works is as follows. So you have your DNA duplex, your target DNA, and you add primers to flank your DNA sequence that you want to enhance or multiply. And your primers are called a forward primer and your reverse primer, and it flanks your sequence of interest. And they must also bind to opposite strands. So here our forward primer will bind to our 3' to 5' strand, and our reverse primer will bind on our 5' to 3' strand. And just think of it as bookends, and everything in the middle of our primers is what we're looking to amplify. And the steps for PCR are as follows. So we add our primers to our DNA solution. We heat our DNA to 95 degrees centigrade. By heating DNA to 95, you break apart all the hydrogen bonds and you take double-stranded DNA into single-stranded DNA. You then cool your sample down to 55 degrees centigrade. At 55 degrees, your primers will anneal to your strands of DNA. You then raise your temperature to 72 degrees centigrade, and for this, Taq, so this is not tall, this should be a Q, Taq polymerase will do all the copying. So a polymerase is an enzyme that adds new nucleotides to DNA. It's what duplicates your DNA. Taq polymerase is a polymerase found in organisms that live in hot springs, so it is heat resistant. If you tried to use a human polymerase or an E. coli polymerase in doing this process, that polymerase would not work because it is too hot. It doesn't work at 72 degrees centigrade, but a Taq polymerase does. Then you repeat steps 2, 3, and 4 about 20 times. So it is a cycle of heating, cooling, then slightly raising the temperature. Heating, cooling, and raising. And every time you do steps 2, 3, and 4 for one cycle, you will duplicate the DNA in your system. And if you do 20 times, your sequence of interest will increase by 1 million times. Right? So if you had one copy after doing PCR, which this process only takes a few hours, you will have 1 million copies. And processes 2, 3, and 4 are all automated. They're automated in a machine that we call a thermocycler. The name makes sense, thermo, heat, cycle, because it's doing these steps over and over and over again. Now, let's say we have our DNA of interest and we put it in our vector, and we've successfully put that vector in an organism, which we covered in previous videos. Now we have means to create a protein of interest, because the whole idea of doing this is that we want to grow a lot of protein in an organism so we can study that protein, right? And so bacteria that have a vector in them, 30% of a bacteria's protein can consist of our gene of interest. And making the protein in that bacteria, we call that expressing your protein. Now, you can study eukaryotic proteins inside of a prokaryotic host. So you can use E. coli to grow mammalian proteins if you want. couple things to keep in mind. One, bacteria and prokaryotes cannot deal with introns. So your DNA sequence has to only be exons. And two, prokaryotes usually don't post-translationally modify their proteins, especially with sugars. Eukaryotes often put sugars on their proteins when they're already made. So if your protein has a sugar tag on it, you can't grow it in E. coli because they'll be missing that tag and may not be able to function well. And you can also do a process called site-directed mutagenesis, and that's what's happening in this photo here, or this picture here. So let's say you have a bacteria with your vector and your protein of interest, which is light green. And you want to study which amino acids are critical for this protein's behavior. And the way to do that is you can mutate the DNA sequences and change which amino acids are being put in your proteins. And that's called site-directed mutagenesis. And the way to do this is that you do PCR with a primer that doesn't match 100%. So here, let's say we're trying to add 3 new bases. Well, when we do PCR, what is going to happen is that you're going to have a DNA mismatch here. And these new 3 bases through PCR, remember what happens in PCR is that these 2 strands will split during our first initial heating phase, and then these mismatches won't matter because our new polymerase, our Taq polymerase will come in and create new DNA sequence. So it will go A, G, C, and then instead of a T here, we will have a G to match our C. Then T, C, A, G, then it will go C, A. So we have effectively changed our DNA code by 3 bases. Therefore, we have created a new, never before seen protein that has different amino acids, and we can start studying that protein. And so just to look at some proteins that have been made using this process, we have insulin, HGH, colony stimulating factors, bovine growth hormone. A lot of different proteins today are grown using this recombinant method because that is the most cost-effective way of obtaining proteins, and we can study how that protein works inside the lab once we have purified copies of that protein. And later on in further videos, we'll talk about how we purify proteins. Now, mice, or any organism that has DNA in it that isn't natural, that humans put in, we call these transgenic organisms, right? And these transgenic organisms have transgenes, which are genes that we incorporate into them. Now, there's various reasons for creating transgenic organisms. For example, we have lab mice if we want to study the effects of proteins in organisms. Or we can also use them in agriculture. So if we want to have plants that have more nutrition or are drought resistance, we can create transgenic organisms, or they're called GMOs. You've probably heard that term before. It means the same thing. Or let's say we want to create livestock that can grow faster with less food or are disease resistance. We can make transgenic organisms. Now, if we want to have our transgenic organism pass on these transgenes, we must place our transgenes in the germline cells. And the way that we can create transgenic organisms, such as mice, is that you have to micro-inject DNA into the fertilized egg. Then that egg is implanted into the uterus of a foster mother, and with luck, that's what it comes down to sometimes. That mother will give birth to animals that have these trans genes. Now, talking about this manipulating of DNA brings us to the subject of gene therapy. Gene therapy is very much in its infancy right now. And the idea of gene therapy is that you introduce genetic mutations with the hope of providing a therapeutic effect. And we have, well, the scientific community has used this successfully already from some genetic diseases. But this is still very risky as diseases are very complex. They usually aren't based on one protein alone. And we are messing with DNA, and DNA is very complex. We don't understand how all of DNA works at all. So using gene therapy, 2 children have actually developed leukemia because of gene therapy. And it's also possible, since we use viral vectors for human gene therapy, that you might have a fatal reaction to the viral vector itself. So there are still many risks for gene therapy, but there is a lot of promise in that many genetic disorders have the possibility of being treated using gene therapy. But again, it is still many decades away from being commonplace and safe, completely safe. All right, and that's it for this video. I will see you in the next video.